Photosynthesis Lab. Purpose: observing the speed at which oxygen is produced in photosynthesis when exposed to different wavelengths of light. Hypothesis: if the disks are exposed to 725 nanometers or red light, then photosynthesis will occur at a faster rate and more oxygen gas will be produced because red light has maximum absorption, which means a higher amount of light will be used. Control variables: temperature of solution, pH of solution, time exposed to light, size of light bulbs. Independent variable: the various wavelengths exposed to the leaf disk. Dependent variable: the amount of leaf disk that float to the top of the cup within 3 minutes. The materials needed include baking soda, a weighing scale, water, liquid dish soap, straws, spinach leaves, a syringe, 250 milliliter beakers, filters of wavelengths of light in the colors green, purple, and red, and a timer. The procedure is: first, mix 0.5 grams of baking soda with 500 milliliters of water to create a bicarbonate solution. Add 5 drops of liquid dish soap to the solution and mix. With the straw, cut out 10 circles or discs from the spinach leaves. Take the syringe, remove its plunger, and also remove the cover from the tip, if there is one. Put the leaf discs into the barrel of the syringe and then tap them down to the tip of the syringe. You can do this by using a straw to gently blow the disc towards the tip. Put the plunger back into the syringe, being careful to not touch or damage the leaves. Pour 150 milliliters of the bicarbonate solution into each beaker. Try to avoid making suds. Draw approximately 6 to 8 milliliters of the bicarbonate solution into the syringe. The leaves should float in the solution. Hold the syringe with the tip upwards and remove the air by gently pushing on the plunger. Plug the tip of the syringe tightly with your finger and gently pull the plunger backwards to create a vacuum. There should be tiny bubbles coming from the disc. Hold the plunger like this for a few seconds before releasing it and letting it snap back. Cause some leaves to sink. Repeat step 11 several times until no discs are floating in the solution. When all the leaves have settled, remove the plunger and pour the disc and bicarbonate solution into the beaker. If any leaves float, remove them from the beaker. Repeat this 3 times separately and make sure there is the same amount of leaves in every beaker. Set up the light fixture so that it's 12 inches above the table. Test one wavelength filter and record the number of leaves that rise within 10 minutes. Repeat for each filter with a new paired beaker. The recorded results were trial 1, red light, 5 floating leaves, trial 2, green light, 0 floating leaves, and trial 3, purple light with 1 floating leaf. Additional notes were that bubbles were seen in the beakers. Discussion. Before analyzing the results, let's first explain how photosynthesis works. Plants convert carbon dioxide into oxygen through their cellular respiration. Plants capture radiant energy to convert into chemical energy to power their needs. This process can be represented with the following equation. There are 2 reactions which make up photosynthesis: light-dependent and light-independent. In this lab, the focus was on how light-dependent reactions are affected depending on the wavelength of light. The wavelengths of light determine how much energy there is. Longer wavelengths have less energy than shorter wavelengths. This is visually seen with different colors, as on our Roy G. Biv spectrum. Red light has the longest wavelengths and purple the shortest. This matters as pigments are compounds that absorb certain wavelengths of visible light, and multiple kinds of pigments are present in leaves. In plants, the main pigment is chlorophyll, which is green. Chloroplasts in plants have 2 kinds of chlorophyll pigments, chlorophyll A and chlorophyll B. The difference between the two is that A reflects bluish greens and B reflects yellowish greens. Since these pigments are green, we know they don't absorb green, but rather they absorb violet-blue and especially orange-red wavelengths. Other pigments exist called accessory pigments. They are, for example, carotenoids like beta-carotene that reflect orange, and xanthophylls which reflect yellow. These are what absorb green light, but these pigments aren't as present in leaves as chlorophyll is. It is known that pigments heavily affect photosynthesis because if the production of oxygen gas, a main product, as seen with the aforementioned equation, was graphed dependent on wavelength, this would be the action spectrum, and it would look very similar to the absorption spectra, showing a correlation between the two. Light, also known as photons, is not only absorbed by pigments. Photosystems are photosynthetic pigments, which are compounds that send light energy to other compounds. Photosystems are located in the thylakoid membrane and have an antenna complex and a reaction center, which are responsible for different functions. The reaction center is made up of a pair of chlorophyll A molecules and proteins, whereas the antenna complex is made up of chlorophyll B and accessory pigments. Plants have two photosystems, P700 and P680, which are named after their special wavelength which they absorb best at. So, to collect energy, photons hit the leaves and bounce around pigment molecules. Eventually, all collected energy goes to the reaction center so that when the center receives enough energy and gets excited, it can raise two electrons to a higher energy level so that the electrons can be transported to an electron acceptor. This is called photoexcitation. So from this, an understanding can be gathered that different wavelengths result in more optimal rates of photosynthesis due to absorption depending on pigments present in plants, as well as the length of the wavelength. Additionally, to explain the addition of baking soda and dish soap in the experiment, this was done to make a bicarbonate solution and provide carbon for photosynthesis. The experiment was measured based on how many leaf discs were floating after a certain amount of time, because this shows how much O2 was made, as oxygen gas produced through photosynthesis would have nowhere to go in the water, thus making the leaves float. Since this experiment gave 10 minutes for the leaves to rise, the amount of leaves which floated also exhibited the rate of reaction, as more energy would be signified by a faster production of O2. In this experiment, though purple would provide the most energy because of its frequency, red light was hypothesized to make the most leaves float, as leaves are mainly composed of chlorophyll pigments. This was proven to be true, as 5 leaves floated for red, 1 leaf for purple, and finally none for green. The green light's results make sense, as green light would be the most reflected light for the green spinach leaves. To conclude, the hypothesis was proven to be correct, and red light is the best for a faster rate of photosynthesis, and consequently, oxygen gas. From this experiment, there were multiple experimental errors. There is one which could have impacted the results heavily, so therefore should be addressed. The spinach leaves used had been sitting in the fridge for a long time before they were used in the experiment. One can tell this by feeling the texture. The leaves were soft, some even mushy and blackened at the edges, showing signs of denaturation. This could have impacted the results in many ways, as it was a struggle to punch them into circles, which could have damaged some leaves but not others. These leaves are questionable as to whether they could have accurately displayed results, even though the results had proven the hypothesis. The fact that no leaves at all floated for the green light was surprising, as leaves still have other pigments in them which would absorb the green light's photons. From this experiment, more leaves were expected to float, but the results were extremely minimal. Furthermore, the experiment is not as applicable to real life if only tested on plants close to rotting, as the only use for this information would be for growing plants. Perhaps, had fresher leaves been used, the experiment could have shown either more extreme results or even different ones.